Hello friends, welcome back to my channel Plant Pathology Pathshala. In this video, we will discuss all about diseases of rice. So everything about rice diseases we will discuss here, all important diseases. This is going to be helpful for you people for basic to advanced level, all type of entrance exams. Okay, so listen this very carefully. Okay, because each and every points are very important. जो इम्पॉर्टेंट पॉइंट है उसको ही मैंने मर्ज करके ये पीपीटी बनाया है. सो एक सिंगल पीपीटी में आपको एक सिंगल वीडियो में आपको पूरा राइस डिजीज़ कवर हो जाएगा. सो विदाउट फर्दर डिले लेट्स स्टार्ट द वीडियो. सो फर्स्ट द मोस्ट इम्पॉर्टेंट डिजीज ऑफ राइस, दैट इज द राइस ब्लास्ट. So as you all know, rice blast is a fungal disease. So the pathogen or the fungus that is causing the rice blast is Magnaporthe oryzae. Okay, so this is the perfect stage. Perfect stage means it produces sexual spore, and sexual spore it produces the ascospore. Okay, it produces sexual spore ascospore, and asexual spore is conidia. And its asexual stage is Pyricularia oryzae. So remember, the pathogen causing rice blast disease is a fungus. Its perfect stage is Magnaporthe oryzae, that is producing the ascospore, and the asexual stage or imperfect stage, that is lacking or not producing sexual stage, not producing the sexual spore, it produces conidia, that is the Pyricularia oryzae. Next, disease type. So it is a polycyclic disease. Polycyclic disease means in a single season it completes more than one life cycles. Okay, by production of conidia. Next, the diagnostic lesion. So the characteristic symptom produced by rice blast is spindle shaped lesion with grey center and brown margin. See, here the photo is there. You can say diamond shaped or spindle shaped or eye shaped or boat shaped lesion will be there. Means at the end, both and these tapering ends are there. Okay, and it is covered by a brown margin, and the central portion will be grayish in color. In this photo you can see here, this is spindle shape or eye shaped with a brown margin and gray color center. Most characteristically it is for the leaf blast. Okay, then I have already discussed in a separate video that the rice blast is consist of three major phases, that is leaf blast, node blast and neck blast. And among these phases, the most damaging is the neck blast. Why? Because it causes white ear, white head. This is the photo of white ear. What happened when the neck region of a panicle is infected, means rotting of the neck region which holding this panicle got rotten. So due to rotting, no photosynthesis will pass from the leaf to the panicle or the grains, so no grain filling will be there. You can say though this ear become white, or it is also called as a blanking also. Okay, and it is also known as rotten neck disease also, because ultimately this neck region is blackened, rotting is there. That's why it is also known as rotten neck disease. Next important point is the inoculum. So the primary source of inoculum comes from infected seed and crop debris. Apart from this, also the collateral grass host like Echinocola, Lurcia hexandra, then other species, other rice weeds are also serve as primary source of inoculum. So primary source of inoculum is seed, infected seed, crop residue or crop debris, and collateral weed host. Next is secondary spread. So secondary spread occurs through airborne conidia. Because of this, it is polycyclic in nature. Okay. And very important point about the conidia is it is pyriform shaped. See, this is the conidia is like this. It is 3-celled, means the conidia is pyriform, pear shaped with 3-celled because of presence of 2 septa. 2 septa are there, which is dividing this conidia into 3-celled structure. Then about the penetration. So we all know that the fungus mostly penetrate by mechanical pressure. So how this fungus is generating that mechanical pressure to penetrate the leaf cuticle? By producing a specialized structure, a specialized pressure bag, that is nothing but the aprosorium. Okay, means actually when it germinate, it will produce when it come in contact with a leaf surface, it produce a pressure bag like pouch like structure where the pressure is accumulated, and then using that pressure infection bag will penetrate the leaf tissue. Okay, this pressure bag is called aprosorium. This is characteristic structure which is very necessary for infection by the Magna Porthoraisi. Remember this, aprosorium. And the most important substance that is responsible for creating this pressure is glycerol. Remember this also. So due to the osmolyte glycerol, this high pressure is created inside the aprosorium. And another substance is melanin. The aprosorium wall actually consists of melanin, which actually helpful for restricting this glycerol, means it is not allowing the glycerol to come outside. So for the pressure maintenance, the glycerol and melanin is very important. Remember this also. Next, about the favorable condition for the rice blast diseases. Temperature you have to remember 20 to 28 degrees Celsius, and then relative humidity more than 90%. This is very important. High relative humidity and temperature and cool temperature, that is 20 to 28 degrees Celsius. Not exactly cool, you can say, just moderate temperature 20 to 28 degrees Celsius, and high humidity, very high humidity is required. If that leaf is not having sufficient amount of moisture, the pathogen will not at all germinate, means the spore will not at all germinate. And then another most important thing, for sporulation, 14-hour light and 10-hour darkness is necessary. See, this question was already asked in 2025 ARS net exam. Okay, so remember this. For the sporulation of rice blast pathogen, 14-hour of light and 10-hour of darkness is necessary. Then about the nitrogen application, excess nitrogen favor the disease severity. Then about the management. So chemical management, for chemical management, azole group of fungicide is very effective, because it interfere with the melanin biosynthesis, which is very important for aprosorium formation. If aprosorium is not formed, ultimately pathogen is not able to penetrate the leaf surface and no infection will be there. So this tricyclazole can be used both as seed treatment or as foliar spray. So for seed treatment 2 gram per kg can be used, and for foliar spray 0.1 to 0.2% spray can be done. Then about the biological control, trichoderma viride can be used as seed treatment as well as seed treatment or foliar spray also. Then best management. See, for most of the diseases, best management practices or the most economical long-term strategy is use of resistant variety. Okay, remember this. Then about the little bit about history part. So this rice blast was first recorded in China in 1637. This is very important. See, in state exams and also in JRF also, this question is repeatedly asked, that the rice blast was first recorded in which country? That is China in 1637. Then in India, it was first reported in Tanjore district of Tamil Nadu in 1980. Then also remember the other names for this rice blast. We have already discussed, it is also known as rice rotten neck. Apart from this, the Japanese name of rice blast is Imochibyo. In Italy, it is also called as bruschone. Okay. So let's revise once again about rice blast. These are the very important point. Okay. So this rice blast disease first reported in China 1637. Other names are Imochibyo, bruschone, then rice rotten neck. In China it is also known as yellow fever, rice yellow fever. Remember this, okay? Then about the pathogen. So pathogen is the Pyricularia oryzae. The imperfect stage, perfect stage is Magnaporthe oryzae, which produce the ascospore. Then it is a polycyclic disease. Diagnostic symptom is spindle shaped lesion with grey center and brown margin. Most damaging stage, neck blast. Primary source is from infected seed and crop residue. Secondary spread from airborne conidia. And how this conidia look like? Conidia is pyriform or pear shaped, 3-celled because of presence of 2 septa. Then penetration occur through aprisorium. Favorable condition, temperature 20 to 28 degrees Celsius, relative humidity more than 90%, and sporulation favored by 14 hour of light and 10-hour darkness. Excess nitrogen favor the disease. Chemical control by using the tricyclazole. Okay. Best management practice by using resistant variety. So this is all about rice blast disease. Next, about the very important disease, bacterial leaf blight of rice. So the causal organism for the bacterial leaf blight of rice is a bacteria, obviously. So it is caused by Xanthomonas oryzae, Pathover oryzae. See, two diseases are there. Bacterial leaf blight of rice and bacterial leaf streak of rice. So bacterial leaf blight of rice is caused by Xanthomonas oryzae, Pathover oryzae, whereas bacterial leaf streak, BLS, this is BLB, whereas this BLS, bacterial leaf streak, was caused by Xanthomonas oryzae, Pathover oryzae cola. Okay, just difference in the Pathover. Here it is oryzae, and in case of BLS it is oryzae cola. Just little difference is there in the symptom. We will discuss it later. Okay, first let's discuss about the BLB. So about the history, it was first discovered or observed in Japan 1884, and the crescent phase was first reported in Indonesia. In India it was first reported in 1959. The causal organism we have discussed, Xanthomonas oryzae, Pathover oryzae. Then about the pathogen or the bacteria, it is a gram-negative bacteria. It is aerobic, non-spore forming and capsulated bacteria. This is very important. Gram-negative, aerobic, non-spore forming and capsulated bacterium. Then about the flagellum. It is monotrichous polar flagellum. Means the bacteria is having a single polar flagella. Okay. That's why it is called as monotrichous polar flagellum. The infection coat means how this pathogen entered. So as you all know that fungus, nematode, they actually use some mechanical energy or enzymatic forces to enter into the plant cell, but the bacteria don't have any such mechanism. It entered the plant cell through natural opening as well as wound. Two major sources are there. So through which natural opening this bacteria enter? So for the bacterial leaf blight of rice, the Xanthomonas oryzae, Pathopora oryzae, it enters through the hydathodes. Okay, this is also called as water pores. तो क्या होता है, वो जो लीफ होता है, लीफ के कॉर्नर में ऐसे वाटर पोर्स रहते हैं. दैट इज नथिंग बट दिस hydathodes through which this leaf, this Xanthomonas oryzae enter into the plant cell. Very, very important question. Okay. Then about the crescent phase. See, this bacterial leaf blight symptoms are actually categorized into two phases. Initially, when it is in the seedling stage, means the rice is in seedling stage, that time the seedling wilt, systemic seedling wilt occur. Sudden systemic wilting of small seedlings, that is known as crescent. So entire nursery will look wilted. 3 to 4 week plants. Okay, that phase is known as crescent. And in the further at tillering phase, we will observe the characteristic symptom like this leaf blighting. Actually, what happened in the this leaf actually gradually start. First, initially water soaked lesion will form, because in every bacterial disease, initially the water soaked lesion will form. Then the yellowing gradually start from the tip to downward. Okay, remember this, from the tip to downward blightening will occur. And the most characteristic thing is the yellowing of leaf, that the margin of this yellow portion is curved margin. Okay, remember this, curvy margin is there. Then mode of infection of this pathogen. This pathogen actually colonizes the xylem tissue. Xylem vascular colonization is there. So this is a systemic infection by colonizing the xylem tissue. Then about the source of inoculum. So source of inoculum, primary source is seed, crop debris, collateral weed host or soil. Then secondary inoculum is the bacterial ooze, that mostly spread by the rain splashes, irrigation water. Okay. Next, about the favorable temperature, 25 to 30 degrees Celsius. Excess nitrogen favor the disease. Then about the resistant gene, see this XA21, this is a very, very important gene that is showing resistance against the bacterial leaf blight of rice. And important donors for resistant gene is tetep, teducan, geneth. Then virulence factor. See, TAL effector is contributing to the virulence factor for this pathogen. Means actually TAL means transcription activator like effectors. TAL. This actually modify the transcription, means gene transcription in the plant in favor of the bacteria. तो क्या होता है, ये एक ट्रांसक्रिप्शन फैक्टर है, एक इफेक्टर है जो बिहेव करता है एक ट्रांसक्रिप्शन फैक्टर की तरह, जो मॉडिफाई करता है प्लांट के जीन एक्सप्रेशन को, और प्लांट के जीन एक्सप्रेशन को इस तरह से मॉडिफाई कर देता है कि अल्टीमेटली वो पैथोजन के फेवर में प्रोटीन्स या फिर अदर थिंग्स प्रोड्यूस करने लगता है. ओके, दैट्स व्हाई दिस टाल इफेक्टर इज कंट्रीब्यूटिंग विरुलेंस for this pathogen. Then about the management, best management practices is obviously use of resistant variety, then use healthy seeds. Then for this chemical we can use some bactericide like the agromycin, tricyclazole, any sorry, not tricyclazole, tetracycline, other antibiotics we can use. Okay, then removal of collateral host, balanced use of fertilizer because excess nitrogen may favor the disease. Then ultimately the integrated disease management for sustainable management. Okay, so this is about the bacterial leaf blight of rice. Let's revise once again. So first observe in Japan, most important and damaging phase is the crescent phase. It occur in seedling stage of 3 to 4 week plant, first reported in Indonesia. It was first reported in India in 1959. Causal organism Xanthomonas oryzae, Pythophora oryzae. The nature of the bacteria is gram-negative aerobic non-spore-forming and encapsulated bacteria. It is having a single polar flagellum that is known as monotrichous. It enters through hydrothodes. Then it colonizes xylem tissue, so it causing systemic infection, systemic wilting it cause. Okay. Then primary sources mostly the seed, crop debris, collateral host, weed host. Secondary inoculum is bacterial ooze. Then favorable temperature 25 to 30 degrees Celsius. Then most important gene, resistant gene is XA21 and virulence factor is TAL factor. Next, about the very important viral disease, rice Tungro disease. This is very, very important. First, see the causal agent. See, it is caused by two viruses. Both the virus complementing each other. Without another virus, a single virus can't produce sufficient disease. Okay, so it is caused by two viruses, rice Tungro bacilliform virus RTBV and rice Tungro spherical virus. So these virus not only different based on its morphology, it is also differ based on its genome also. Okay, so this RTBV, rice Tungro bacilliform and rice Tungro spherical virus, both complementarily causing this rice Tungro disease. So this rice Tungro bacilliform virus, it causes major symptoms, whereas this rice Tungro spherical virus, it assists transmission. So if only the spherical virus is present, mild symptom will be present. But if both this virus are present, means both this RTBV as well as RTSB are present, then we will get major symptoms. But when it is the plant is infected only by rice Tungro bacilliform virus, then it will produce symptom in that plant, but it can't transmit. Okay, तो जो क्या होता है, राइस टुंग्रो का जो स्पेरिकल वायरस होता है, दैट इज फॉर द ट्रांसमिशन. अगर राइस टुंग्रो बैसिलिफॉर्म वायरस का इन्फेक्शन है तो हमें प्रॉपर सिम्टम्स मिल जाएंगे, बट वो एक प्लांट से दूसरे प्लांट ट्रांसमिट नहीं हो पाएगा. फॉर ट्रांसमिशन इट नीड्स द स्पेरिकल वायरस. देन अबाउट द जीनोम ऑफ दिस टू वायरस. सो राइस टुंग्रो बैसिलिफॉर्म वायरस जीनोम इज सर्कुलर double stranded DNA, whereas genome of rice Tungro spherical virus is polyadenylated single stranded RNA. So remember this is very important for RTVV double stranded DNA and for RTSB single stranded RNA. Then bacilliform particle means definitely the shape is bacilliform in it is bacilliform in shape and its size is 130 to 30 nm. 130 into 30 nm, and for spherical particle it is spherical in shape with diameter 30 nm. So characteristic symptom is yellow to orange discoloration. Here the symptom are there. So it produce yellow to orange discoloration of the leaf. So yellow color discoloration mostly observed in Japonica cultivars, whereas orange color discoloration mostly observed in Indica cultivars. And this yellowing or orange discoloration started from the leaf tip. Remember this. And to identify the virus, iodine test is done. So actually iodine test most done for the starch accumulation. So this virus actually due to infection it accumulates starch. So by iodine test we will get dark blue streak if the virus is present. Then other symptoms are reduced tillering and stunting, and infected stubbles act as inoculum source. Then about the vector. See, vector is very very important. You remember the scientific name as well as the common name. Nephotetics virescens. This is the GLH. Okay, green leaf hopper. Nephotetics virescens. This is the vector for rice Tungro disease or virus, and it transmits the virus in a non-persistent manner. Remember this also. And the transmission of virus is assisted by RTSV. This RTBV can't transmit alone. And management practice for this is use of tolerant cultivar, and for most of this viral disease, vector control is most important. So about the rice Tungro disease, most important point is it is caused by two different viruses. Rice Tungro bacilliform virus, which is having double stranded RNA, bacilliform safe, and it is actually and its size is 130 to 130 into 30 nm. And another particle is rice Tungro spherical virus with spherical safe containing single stranded RNA as genome, and size is 30 nm diameter. Most important test to detect the virus is iodine test. Then characteristic symptom is yellow to orange discoloration started from the tip. Then it also causes reduced tillering and stunting. Major vector is Nephotetics virescens. Transmit the virus in non-persistent manner. Management is use of tolerant and tolerant cultivar with vector control. So any type of insecticide you can apply for the vector control. Next, about the very important emerging rice disease, newly reported rice disease in India, that is rice black streak dwarf virus. See, this was first reported in Japan, and actually two variations are there: rice black streak dwarf virus and southern rice streak dwarf virus. This southern rice streak dwarf virus actually reported in India, which was previously reported in China. But this rice streak dwarf virus which was first reported in Japan, was not reported yet in India. Then the family of this virus is Spinaerioviridae. Then genus is Physiviridae. The genome is 10 double stranded segments. See, the virus is multipeptide virus which contain 10 double stranded RNA segments. Vector very very important, WBPH, white backed plant hopper. Remember the scientific name as well as this common name, the Sogatella furcifera. Transmission occur in persistent propagative manner. See, for most of the plant hopper, it transmits viruses in persistent propagative manner. Remember this point. Then the major symptoms include stunting, black streaks, and excessive tillering. It is not transmitted by seed, and best management practices is vector control, roguing, insect-proof nursery. So this is about this rice black streak dwarf virus, first reported in China. It is recently reported in 2003 in India. It is having 10 double stranded RNA segments. Remember the vector name, WBPH. Its scientific name is Sogatella furcifera. Transmission occur in persistent propagative manner. Okay, this point are very very important. Next, about the brown spot of rice. The pathogen or the fungus causing the brown spot of rice is Bipolaris oryzae, which is also known as Cochleobolus miavinus. Its old name is Helminthosporium oryzae. Then it is a seed-borne and airborne disease. The characteristic symptom is brown spot with gray center and brown margin. See, in case of brown blast, spindle shaped lesions are there, but here it is oval or oval shaped lesions are there. That is the main difference. Otherwise, it is also with brown spot with gray center and dark brown margin. Okay. And another thing is characteristic yellow halo can be seen here in case of brown spot, but that characteristic halo is not seen in blast mostly. Then another important historic event related to this disease is this is responsible for the Great Bengal famine of 1942 to 43 that caused death of 1 to 3 million people due to hunger. Okay, then favorable temperature for this disease is 25 to 30 degrees Celsius. Best management practices use of seed treatment with carbendazim 2 gram per kg because it's a seed-borne disease. Then other important fungicide that can be foliar spread are mancozeb, propiconazole, carbendazim, hexaconazole. Okay, so this is about the brown spot of rice. Remember, it is the reason behind the Bengal famine. Then about it is seed-borne. Its pathogen you have to remember. Then about seed treatment, its dose, and about the foliar sprays. Next, about fall smut of rice. The pathogen, this is a fungal disease. It is caused by Velociclava virens. See, this is the new name for this pathogen, Velociclava virens. Older name is Astylagonoide virens. Then characteristic symptom. See what happened. The individual grain, this rice grain actually converted to greenish yellow velvety smart balls. Initially it is yellowish orange or greenish yellow in color. Later it turned to black color. Okay, here I have given here how this smart ball progress. As initially they are greenish, then little bit yellow, orange type, finally it become black. Then the infection stages, the booting stage of rice. Then primary source of inoculum is ascospore on the sclerosia. Secondary inoculum is the chlamydospore. It affects only few spikelets. High humidity favor the disease. Excess nitrogen increase the disease incidence. And in historically people actually believe that farmer actually believe that it is a good sign of harvest. तो क्या होता था, पहले लोग सोचते थे कि ये ऐसा अगर कुछ मतलब जो राइस के पैनिकल में ऐसे वेलवेटी ग्रोथ आ रहा है तो हमें अच्छा खासा एक ईल्ड मिलेगा, बट दिस इज ए मिथ. It is a disease. Okay, small ball enlarge and change color with age. Here it is mentioned. It is associated with a mycotoxin, that is osteoloxins. Then protectant fungicide can be used, that is copper oxychloride. Timely spray at booting and panicle emergence can control the disease. Then best management practices, use of resistant variety and IDM practice. See, क्या होता था, हाई ह्यूमिडिटी फेवर द डिजीज़. तो जब पैनिकल फॉर्मेशन का स्टेज होता है, जब फ्लावरिंग का स्टेज है, उस टाइम अगर थोड़ा सा बारिश होता है तो हमें ये डिजीज़ का इंसिडेंस ज़्यादा मिलता है. और ये अभी जब अगर अच्छा खासा ह्यूमिडिटी प्रेजेंट रहता है तो प्लांट का ग्रोथ भी अच्छा रहता है. तो इसीलिए लोग जनरली सोचते थे कि दिस इज़ ए गुड साइन ऑफ हार्वेस्ट, बट दिस इज़ नॉट लाइक दैट. दिस इज़ ए डिजीज़. It cause yield loss. Okay, so remember these points. These are very, very important points. Then about the sheath rot of rice. Sheath rot is also very, very important because it directly affect the grains. Okay, so it is caused by fungus Sarocladium oryzae, first described in Taiwan 1922. It also affect the booting stage of rice. Then the boot leaf encloses the panicle is affected. See, this is the boot leaf that enclose the panicle. When it is affected, it ultimately affect the panicle and ultimately our yield is affected. So white powdery fungal growth inside the leaf sheath. So conidia is hyaline. Then cylindrical is single celled. It is seed borne and airborne. High humidity favor the disease. High humidity favors means then obviously dense canopy increase the severity. Then brown plant hopper injury predispose the infection. Then mild injury also favors the infection. Then it can cause panicle sterility, produce chaffy grains. Seed treatment is useful with carbendazim 2 gram per kg you can use. Then best approach is obviously IDM. Okay. Remember this causal name. It affects the booting stage. Then about the seed borne nature, high humidity. Okay, these are important points. Now, next about the stem rot of rice. This is also very important. It is caused by Sclerotium oryzae, the fungus. Its sexual stages Leptospirea salvini, first reported in India in 1913. It is considered as a wound parasite. Okay. Initial symptom are small black lesion near the water line. Here you can see small black lesions are formed near the water line, and the most characteristic sign is presence of the sclerosia. See, this type of sclerosia can be seen in the infected portions. Then lodging is characteristic consequence because it is affecting the stem. Ultimately plant will fall down because the culm tissue or the stem lesion become rotted. The primary source of inoculum is soil and crop residue. High humidity favors the disease. Excess nitrogen also increases the disease severity. Stem borer injury predispose the infection. And the deep summer plowing helps in reduction of inoculum because here the pathogen or the fungus is producing sclerosia. This can be killed by deep summer plowing. So here we can use the deep summer plowing. Field sanitation is also important for removal of debris or the sclerosia. Then best management practices use of IDM. So this is about the stem rot of rice. Then about another important disease, sheath blight. This is also a very important disease caused by a fungus, Rhizoctonia solanae. Its sexual stage is Thanatophorus cucumeris. Then it's a basidiomycetes fungus, and initial symptoms include greenish to grey oval lesions. Okay, on the leaf sheath, here you can see greenish to grey oval lesions will form. Most susceptible stages tailoring to heading stage, favorable temperature 30 to 32 degrees Celsius, and favorable relative humidity is more than 90% you can write. Then survival structure is also here, the sclerosia. So same management practices can be applied like deep summer plowing. See, wherever sclerosia are present, it is the secondary spread are through the irrigation water, whereas the primary source of inoculum is the sclerosia, which is present in the crop debris or soil. Then host range is more than 188 species. See, this question mostly that which pathogen is with very wide host range, that is Rhizoctonia. Excess nitrogen increase the disease. Dense canopy also favors the epidemic. Biocontrol use for this pathogen is Pseudomonas fluorescens. Then lesion coalesce and cover the sheath. Ultimately, blightening of the sheath will occur, like severe leaf blightening occur at later stages because of coalesce of this lesions. Best strategy is IDM for the management. Okay, about this sheath blight. Next about very important few less important, but from the exam point of view these are very important. See, you have to remember the causal name and vector for these viral diseases. First about the fungal Bakanae disease. This is actually this is a fungal disease. So from this disease infected plant, the gibberellic acid was isolated. So this Fusarium fusicoroi or the Gibberella fusicoroi, this produced the GA3, and this is responsible for this GA3 is responsible for foolish elongation. What happened when this disease occur in plant? Excessive elongation of seedling occur. That is due to the presence or the due to production of GA3 by the fungus. Okay, that's why excessive elongation is observed. So for this disease, you have to remember the causal organism, and the major symptom is excessive elongation of seedling. The seedling become pale yellow. Okay, and the reason behind the elongation is production of GA3. Remember this. And survival by crop and crop debris and seed. Favorable temperature 25 to 28 to 35 degrees Celsius. So as it is seed borne, so best management practices, use of healthy seed, as well as you can go for seed treatment. This much is sufficient for the Bakanae disease. Next, about the major viral disease, that is rice grassy stunt. So it is caused by rice grassy stunt virus. Most important is its vector. It is transmitted by BPH, Nira Parvata lucens. Remember the common name as well as the scientific name also. Then transmission is propagative, persistent propagative. See here, for the rice grassy stunt as well as rice black stick drop virus, the vector is BPH and the transmission occur through persistent propagative manner. But in case of Tungro, it is transmitted by GLH, the nephotetics virescens, which is transmitted in non-persistent manner. Okay. Major symptoms include severe stunting, excessive tillering, and grassy appearance. The name only suggests here the virus disease name only suggests that grassy means excessive tillering is there. Stunt, it is written means definitely it is causing stunting. See, most of the viral diseases cause yellowing, excessive tillering, stunting. These are very common symptoms for the most of the viral diseases. And best management practice for the viral disease, vector control and use of resistant variety if available, or you can go for tolerant cultivars. Next important viral disease, phytoplasmal diseases, rice yellow dwarf, very important. Okay, this is caused by phytoplasma, not virus. We will discuss what is phytoplasma in separate slide. Okay, these are actually bacterial like organism, but it lacks the cell wall. Okay, we will discuss it. Then the vector is green leaf hopper nematode species. Transmission occur in persistent manner. Then about symptom, yellowing, dwarfing, profuse tillering and sterility in plant. This is the common symptom for the phytoplasma, yellowing, dwarfing, profuse tillering. It is not seed transmitted. Management is roguing of infected plant and vector control. See, whatever disease which are transmitted by any vector are for that disease, best management is control of vector. Okay. Next, the last post harvest diseases includes the contamination with some that actually post harvest diseases are caused mostly by Penicillium species and Aspergillus species. Remember this, post harvest fungus that is causing post harvest disease are mostly the Penicillium species and Aspergillus species. It produces the Aspergillus flavus is known for the production of aflatoxin. This Aspergillus ochraceus is known for the production of ochratoxin. This post harvest diseases are mostly occur due to the improper storage condition, like due to grain, if the grain moisture is more than 13 to 14%, and if high humidity is there, then this post harvest disease generally occur. So best management practices is dry the grain below 13% moisture, proper cleaning and insect free storage, airtight storage. So proper storage you have to do to manage the post harvest diseases. Okay, so this is all about rice diseases, important rice diseases. Few points I have mentioned, these are very important. Don't skip any point. All these points are very important. Okay, so if you have any doubt, please let me know in the comment section. So I think the rice diseases are now clear to you people. So thank you for watching.